So yesterday we have just started with how better can we plan for our teaching so that the students will get the maximum benefit from us. And today onwards, we are going to focus on the execution part. How better can we execute teaching biology so that students will utilize us maximum and the maximum clarity will be attained and maximum preparation for their future will happen. For that, what we are going to see today is that execution part mostly we are going to see. As teacher, yesterday we have taken the example of photosynthesis in higher plants, and today we are going to take the example of respiration in plants. That is a topic today we are going to revise. How better can we execute? As we always say, it is not just a teaching methods. It's like we are all discussing with each other. So your valuable inputs also very important for us. So kindly make your inputs also together. We'll learn, we'll execute, will improve our areas. So we have studied about, we have discussed about various advantages of making a lesson plan. A lesson plan is a very important material that too as of today, the current scenario, current ongoing pandemic, we have to be well planned. As already we said, when we test the previous knowledge of the children, there will be a very large, very huge gap that from those who are coming from 10th standard to 11th standard, definitely there is a huge gap already that we need to plan our lesson in such a way that the gap can be filled as much as possible. Second thing, the students motivation or that part also very very important, and evaluation, all these parts we need to give little extra care than what we used to do previously. Compared to that, we need to give little more extra care so that in a very better way, all together we can reduce the learning gap as much as possible. That is it. Same way, the individual, the curriculum is going to be same for the entire school, all over the school, CBSE school across India, even the some schools which are located abroad. For the entire school, the curriculum is going to be same. The reference material, the NCERT textbook is going to be the same. So everything is going to exam pattern. The end of the term board exam is going to be the same. But what makes the difference of each and every school and each and every students, those who are going to participate in the board exam, is how we prepare the individual teacher's methodology, teaching methodology, or the teacher's effort, or what are the methods and how interesting and how in a better way I'm going to teach my students. That is what the students are going to be prepared for the writing the board exam. When everything is same, what makes the difference in the among the student is the teacher who is going to teach only is going to make the changes that is going to happen in the children. So we need to keep that in mind, and we need to take a extra care while preparing the lesson plan. And how, what are the good lesson plan, the care principle of a good lesson plan if you want to look into. So it should not be so very complex at the start itself. It must always start with the simple. Whenever we are executing our work also to the teachers, students, when we are teaching directly, we cannot start away with the direct today we are going to start respiration in plant, respiration is so and so. So we cannot directly involve in the complex concepts for their level. We have to go from very simple idea where the students must travel along with that. There are some places where student will have difficulty, where they cannot continue in a constant manner, where we need to help them how better along with us we can take them. But always the planning should happen in such a way that very simple to the complex only it should happen. And straight away, that's the reason why we say that they also the abstract concepts and all we cannot directly start. So it has to be come from the this concrete to abstract. So very small part to the whole part, not entirely the whole part. We cannot, definitely we cannot complete. When our planning itself, we need to keep in mind everything from a simple part to the whole or to the complex. We have to make it. And we must be very clear about whenever we are giving a lesson plan, and we must be very clear about what is our objective and what is the learning outcome we are expected from the student by learning this particular topic. That is very very important. And while planning itself, we need to also decide what are the other aids. It's not like teaching aids, a chart or anything. It's about even our practical or a practical material that we can brought into the class and we can show it to the children. So what are the teaching aids which is readily available, which is definitely possible to show to the children? That has to be planned well in advance along with our planning itself. And the whatever the activities we are going to do, then before itself, 1 day before, 2 days before itself, we need to prepare what are the activities we are. At the time, we should not be trembling in front of the students. We should not be finding what to be done, where to be found and all. Everything must be readily available so that smoothly the class can be conducted. So these are all the few things that we need to be very clear about when we are planning our class. It's not about lesson plan. We are writing and submitting and getting signed from the principals and we are submitting. It's not like that. It's really a very, this is the only place where our individuality is going to be reflected with the teaching learning experience. Otherwise, all the other things are going to be same for the entire country. The students, CBSE schools, all the students are going to be having the everything same. Everything is going to be the similar only. Only this is the only area which is going to be different, where we need to prove ourselves very well in achieving that. So when we come to the what are the different skills we can use it while teaching the biology, we can know already. We know that we need to introduce the topic, we need to explain, we need to in between, we need to question them, and we need to close the topic. And also reinforcement, very important for the students to motivate. Negative reinforcement I would never encourage, only the positive reinforcement, which is either verbal or non-verbal. How can we reinforce the students to learn it in a joyful manner? So these are the things, generally the skills which is required by a teacher while executing what they have planned. So previously we need to plan in a better way. Second one, we need to execute what we have planned. For planning, we have taken an example of this photosynthesis in higher plants. And now for execution, we are going to take another lesson, that is that respiration, the lesson. So now we all need motivation, isn't it? Teachers also need motivation. So though if the teachers are there on the video mode, then I am going to play a video for you all. It's very interesting video for the children to understand the microscopic level where photosynthesis, how better it happens. So I request all the teachers to be on the video. The audio is not required here only. It's a background music only. There is no explanation. So The video which you have seen is just about the nanometer scale where that photosynthesis is taking place. So you can see a very small light color, green color, the components, they are all nothing but they are the photosystems. So you know very well where it is located. So that is that video. This will make the children to understand how in the very nano scale, we know we send her over to the Mars, we send her over to various different far located planets, and the students generally have the interest to learn about it. But with the cell which is located within the plant where the photosynthesis is going to take place, the students must also understand about the nanometer scale of what is happening. This will motivate the children to learn very deeper and deeper of the plant system. So they must understand what are the biochemical reactions are taking place, how those chemical reactions are taking place, the marvels created by the nature. To understand about how the chemical reactions are taking place is a very important way to motivate the children to learn scientific knowledge. So with this, shall we go to this next topic, teachers? Are you ready? Yes.
Yes, sir.
Yes. So now I'm going to show a slide. By seeing that, you are going to find what is that all about? What is the topic? Yes, teachers. Can anyone define respiration?
So respiration is a breakdown of complex food substances into simpler one where energy is released.
Very nice, ma'am. Very good. Thank you. Yes, teachers. Others also. I request others input also. You can even put it in the chat box, not an issue.
The releasing of energy from the food sources.
Very nice, sir. Thank you. Thank you so much. But now students will be asking question, plants are not eating the food, then how come the energy is released from the food? So if the question asked by the children, how can we answer this question, teachers?
Food is not taken by the plant. We cannot say that. Like all the essential nutrition for the plant is taken up by through the water from the root, and those food materials has to be broken down into simpler one for the release of energy. Similar process only is taking place in us also. Like we take food through the mouth, but food is reaching only the stomach, and from there only all the foods are getting digested, and then it is used for respiration. And that respiration, what we speak here is cellular type of respiration.
Very nice, ma'am. Very nice. Yes, yes, of course. So in the case when we say about plants, plants of course do not utilize the food, do not eat the food, but they manufacture their own food. So here we need to highlight to the students that plants makes the food for their own energy requirement. Whether the carrot, they store the food in the root system, or the potato, the stem, whatever the energy is stored there, that is stored for the plant to utilize for later for respiration. But we are all exploiting the plants and we are taking those things for our requirement. That we need to very clearly to highlight to the students, so that the students will understand what will happen to our life without those plants. If the plants are not doing that photosynthesis and respiration, what will happen to our life, how overall we are exploiting it, and how better we can stop exploiting, we can just in a sustainable manner, how can we use it, that we need to highlight. So thank you so much. Now we'll go on to the next topic. Introduce with the respiration in the plants. That is a lesson today we are going to study how well we can execute this particular lesson. So first, before we start, you know these things about the structure. We need to explain to the children the structure where the respiration is going to take place. While we are explaining the structure itself, while we are explaining about the respiration equation itself, we also need to compare that equation with the photosynthesis equation. How the when photosynthesis, how it is happening, that equation, we need to correlate with this equation of respiration so that the students will be able to understand the difference between the anabolic and catabolic reactions, the overall metabolism, how the anabolism is taking place and catabolism is taking place. They will be very well able to understand, so that we need to, in this point itself, we need to link that photosynthesis also and the this respiration also in this case itself. Of course, respiration is not all exclusively a catabolic process that we have. We have the amphibolic pathway. But here, what is happening to glucose is a breaking down. That when that we are explaining to the children, we also must link that concept with the whatever they have learned in the previous lesson, the equation of photosynthesis. Next one. So respiratory substrate. They are saying we are eating idli, we are eating dosa, lunch, we are eating various food. All these foods are nothing but the respiratory substrate. And we can even explain them with a small diagrammatic representation of two very important organelles involved in the plant system. That is the chloroplast as well as the mitochondria. We can link them very well with each other. And this is the area where we also need to highlight the difference between the photophosphorylation and oxygenic phosphorylation. Both the things we need to highlight at this point, so the students will be able to very well understand not only this particular lesson, even they will be able to understand, link with the previous lesson what they have learned, that also they will be able to connect with that, so that when they make it the connection between this lesson and that lesson, they will very well understand how that photosynthesis happening also, how well that respiration happening also. Both the topics they will be able to correlate and they will be learn it in a very effective manner. Now a quiz time. You're all going to put it in the chat box, the answer for the question. Now I'm going to ask. It is to check in between how active all of us. Yes, teachers. Can you give the answer in the chat box? So next we are going to see the continuation of this. How this respiration in plants takes place. We can explain with that. Already I said we can connect it with the photosynthesis. In this point itself, we need to do that so that students should understand the end product of the photosynthesis is whatever produced that is going to be utilized by the plant for its own energy. That the students must be able to correlate then and there itself. So now this energy currency in this point, ATP, why we consider ATP is a very important energy currency. What is ATP? How the ATP is synthesized here and also in the photosynthesis, there how it is synthesized. What are all the ATP energy currency concept we need to explain in this context itself, teachers. Next one, we are going to study about why that breathing. So here we need to explain about what is respiration and what is breathing. When we talk about breathing, that plants do not have any well sophisticated organ or any particular organ for breathing purpose. And students will also have the doubt, the energy requirement of the plant is very, very low. They are not moving, they don't walk, and they don't do any much work as animals are doing. Then what is the need of respiration? That concepts also will be there. And we need to explain between the woody plant and the stem, the flexible stem plants, both the green plants, both we need to explain what are the different ways their respiration will take place, in what are all the way the exchange of gases will take place. Here, students will get a concept that all the oxygen from the outside, everything is taken by the plant. It's not like, mostly the plant is depending on the internal oxygen level itself. And beyond that, a very less amount of oxygen is consumed by the plant. That also we need to highlight here itself. Yes, teachers. Now your turn. You are going to discuss what is the why there is no any organ for breathing. Yes, teachers.
So because
It's
each part they'll take care of their own gases extension.
Very nice, sir. Yes, sir. Thank you, sir.
And also they are directly in contact with the atmosphere.
Very nice. Yes, everybody can put your input. It's a discussion only, please. Yes, sir. Parthiban, sir. Very nice.
Gas exchange, sir.
Yes, gas exchange. That's what how the gas exchange. Why there is no breathing organ? Why there is no any separate organ for this purpose?
All the parts in the plant, they too respire, sir, but not much amount of oxygen will be carried. Root, stem, every part in a plant will
Yes.
respire, sir.
Yes,
And they may be taking a little amount of oxygen.
very nice, ma'am. Yes, sir and ma'am both have replied very. So this that they are depending on their own. The each and every part will act as the diffusing part, and the diffusion will take place all over the parts in a different mechanism. It's not like one lungs will be pushing the oxygen throughout the body, or a heart will be pumping it. There is no such a mechanism is required, because individually each and every part is there directly in the contact with the environment, and it will be taking the oxygen through diffusion. That is the reason why we need to highlight that very in a elaborated manner. Students will definitely have that confusion. How come the exchange of gases happening? This doubt the students will 100% will have. We need to explain that in a very correct manner, so that students will have that. This next one.
Parthiban sir has posted the answer, sir.
Yes.
Not great demand. There is
Very
no
nice.
great demand.
There you go. Yes, when the students are asking, very nice, sir. That when the students are asking why there is no organ, there is no separate organ. Yes, they don't have any demand for much gaseous exchange, and that's the reason. Very important point, very valid point. That also we need to highlight.
Another important point, sir. Most plant parts are dead. Dead tissues don't require more energy, so the energy requirement is less. The
Yes,
essential
ma'am.
living organs are all in contact with the surrounding. Most part of the plant is dead. Dead tissues don't need any energy. 10th class they would have learned, but we need to
Yes, ma'am.
recall, sir.
Yes, we need to recall that. Yes, we need to recall that also. Yes, ma'am. Thank you, ma'am. We need to recall that also in the 11th standard once again, so that they will understand. So again, that in a completely combustion, here we need to little relate that respiration with the combustion also. How that combustion can be correlated with this particular respiration also. We need to little put an idea to the children, so that they will have some clarity on this concepts. And also the how what is the ATP synthesis here, what it is going to be happen, how it is better it is going to be utilized, that we need to say. So after saying all this introduction, after giving all this idea, then they will be able to understand what is the need of that glycolysis cycle, respiration, all this part. Later they will be very easily, they will travel along with us. And here also we need to say about what is that different types of respiration usually will take place on the basis how the organisms are divided. So aerobic and anaerobic organism, they know. The same time there are some organism that is facultative and obligate anaerobes, and aerobes both are there. Here we need to highlight what do you mean by that facultative and what is that obligate anaerobic organisms and aerobic organism. We need to here itself we need to give a clear definition to the children, so that they will be using that idea in their whenever they are learning the concept in the forthcoming topics, they will be easily able to learn that. Now we are going to enter into the very important topic of this glycolysis. Shall we all enter, teachers? Are you ready? When we are saying glycolysis or breakdown of glucose, when we are explaining, we need to explain it's a very important topic, as we all know that very important in for exam point of view also. It is very important even for the forthcoming years. If they are going to study in college with the biology, they are going to continue, and there also it is a very important topic. They must learn it very well. So it's very important how better way we can make them understand this particular topic. So before we start, what are the challenges have you faced while explaining this concept? You can put forward, teachers. You can either send it in the chat box or you can say it in the orally also you can say.
So one thing, a lot of enzymes are involved, sir. Keeping
Yes,
the
sir.
enzymes names details.
Very nice, sir. Enzyme names, lot of enzymes name their role. Yes, sir. Very nice, sir.
And more than understanding part, it is just like we have to make them memorize everything, like glucose 6-phosphate, everything they have to keep it in memory, because we cannot change this product on any account. So it
Yes,
just
yes, ma'am.
like we have to memorize we have to make them memorize the thing, and it is the very greatest challenge I face when I take the class.
Yes, yes, ma'am. Memorizing. Yes, we need to make, yes, some area we need to even make them memorize the steps and all they have to memorize only. Yes. Any other teachers? Okay. So that's as sir said and ma'am said, the enzymes name, they have to keep it in the order. What are the enzymes are playing there? And second thing, what is the steps in the sequential order? The 10 steps also they have to keep it in the correct order. They have to write that also they need to extra care has to be taken while they are learning and while we are teaching this glycolysis cycle. So before that also there are some doubts and some areas where we need to give equal weightage, that students will very clearly they will say glycolysis is aerobic respiration. They will close their eyes and they will say, but glycolysis is also it's an absence of oxygen, and the anaerobic way it takes place in the cytoplasm where oxygen is in an independent manner, they are it is going to take place. So that also again we need to put that in a very clearly into their mind, because they will very well understand glycolysis for aerobic respiration. That we need to once again highlight to them. And a correct differentiation between a distinction between what are all the events are taking place in this cytoplasm and what are all the events are going to take place in the mitochondria based on the cellular requirement. It has to be clearly put into their mind where is the cytoplasm role and where is the mitochondrium plays a role. They need to understand it in a correct way. Then the scientist name, most of the time what happens, of course we teach them the concepts of what is that glycolysis and everything we teach, but the credit has to go to that scientist also with a lot of effort. With in a later part we are going to discuss that also, how with a lot of efforts with a very limited resource or limited technology the scientists were able to discover all this kind of thing, how they are discovered. So the students must understand the role played by the scientist in a very well, though it is not given in the NCERT reader, those not given in the book. We need to definitely highlight the role of the scientist in a very elaborate manner, in a very excellent manner, because they should know, they should give the credit to the scientist also. That also we need to give little care we need to take while we are explaining about this glycolysis. These three scientists separately, they can even make a project, or they can make even we can make them to conduct some seminar on that. That also we can make them do. Next, this event sometimes misunderstood as it is also a part of glycolysis, but it is not a part of glycolysis. It is a previous part. If the ready-made glucose is available, it's okay. If it is not available, when that in the plant, when it is stored as a sucrose after the photosynthesis, that has to be converted into glucose and fructose by using the enzyme invertase before entering into this glycolytic pathway. Here what will happen, the students will have a understanding about the structure between these two glucose and fructose. That also we need to say. Before saying, I want you to please try to answer for this question. You can put it in the chat box also.
3 carbon compounds. Sir.
3 carbon compounds.
Pyruvate.
Very nice.
Pyruvic
Yes.
acid.
Two molecules,
yes.
two molecules of 3 carbon compound. Pyruvate
Yes,
acid.
very nice.
Pyruvic acid.
It's a yes, 3 carbon molecule. The 6 carbon molecule is split into 3 carbon molecule, which is in 2 in number. Yes, very nice. And also we need to hear how come this glucose and fructose, they are easily using isomerase, we can convert the glucose into fructose or fructose into glucose. That we need to explain to the children in what are the ways they are looking. Both are having C6H12O6 only is the formula. So what is the difference between that two molecule? We need to explain to the children so that they will not have any doubt about the glucose and fructose, and they will be able to learn it in a correct manner that we need to put it. So next, from onwards, the stepwise we need to highlight from the beginning. Even you can give a step numbers. We can usually I do that to my students. The step
Yes,
number will
yes,
be
I
allotted to
also,
them.
sir.
Yes, yes, ma'am. Very nice. So we need to allot the step number to the children for the easy keeping. When we are even conducting the test and all, no need to directly write the entire thing. We can ask them to write the step number 6, write step number 7, write step number 8. Like in a selected steps, if we can give them test and all, if we conduct, then student will be able to recall and keep in the mind that particular step in very well in the mind. So when they know the each and every step know very well, there won't be any problem in putting them in a proper sequence and writing that in during the exam. It won't be a very big issue. So we need to give a step number and we need to make them understand that particular. So here also on the PowerPoint also, the number is given, and you can even make them write correctly, though it is not given in the book. Wherever that ATP is utilized, there they can write -1 and they can circle it. Wherever the ATP is produced, they can write +1 and they can circle it. In that, if they do, in which step, say for example, in write they explain that step 3. If you are giving, while they are writing, they must write that also, and also they should write what happens to the ATP, it is utilized or it is spent or produced, or whether they have to write +1 or -1, they have to write. By writing, the students also will be able to keep the entire step very well in their mind. So we need to give more importance. In that way, we can make the student to teachers. You can also put your inputs. If you have any doubts in between, you can definitely feel free to either in the chat box also you can write it.
Excuse me, sir. Is there any shortcut to remember this all the steps, sir?
Sir, we had one mnemonics for that, sir. I have tried that using of that mnemonics, but it is not much working out. Even if they write that mnemonics, it is not giving a student still facing difficult. Instead of memorizing the mnemonics, they feel like they will memorize this structure itself. They said, still, if there is anything, that also I will share it with you whenever.
Sure.
Thank you, sir. Thank you. Yes.
Sir, I actually, when I teach this one, I convert this into 2 steps, like energy utilizing steps and energy
Yes.
releasing step.
Yes.
So I keep
Very
first
nice.
both, and I keep it as like, see, if you read that, it is just like a rhyming word. Glucose, 6-phosphate,
Yes.
fructose, 6-phosphate, fructose 1,6-phosphate. So when we say that in a rhyming
Very nice.
process,
Very nice.
they are able to understand little bit, like they can keep it in memory.
Very nice, ma'am. Yes, exactly, ma'am. Yes, that's what I said. The part way, instead of going with the full flow, the part ways we can divide. As ma'am said, like, first half and second half we can divide, or we can divide into three steps, or two steps also we can divide. And for that, we may make them to memorize first and make them keep in mind. Then the entire large picture will be there in their mind. Naturally, it will be there in their mind. If they know the small steps, if they know very, very well. Very nice. Thank you, ma'am. Thank you so much. The same way, here we need to highlight that the students will mostly have the confusion about the 4th step and 4th, 5th, 6th, and 7th. These 4 areas we need to again highlight with the proper the same repetition, again and again repetition. We can highlight those topics so the children will be able to keep the topic very well in their minds. This is the very important thing about glycolysis. And the last step is very easy. Anybody, usually the student, those who don't write that first few steps also, the last 8, 9, 10, they will write very well. Because that is not a much difficult one or easy to keep in mind also. So they will write it. But throughout this entire steps, we need to highlight the number of carbon molecule after the 4th step onwards. clearly they have to maintain the number of carbon molecule. That number of carbon is a very important, and how many molecules are produced, that also they need to write it. Right from the test onwards, in the beginning itself, if we highlight and if we reduce the mark, if the students are not writing all this thing correctly, we need to reduce the mark, so that, and also we need to inform the children what mistake they have made, and they won't repeat it in the upcoming exams when we do that. So next one, the EMP pathway, the simplified, or the in one, this brought into one slide for our understanding. Of course, we need not to tell this to student, otherwise students will get confusion about it. It's for us to just to keep in mind. We can use this also a step for keeping in the mind. So now the question time. Teachers can put the message in the chat box. The sudden change from the anaerobic to aerobic process makes some effects. What is that effects called as? The answer is the Pasteur effect. It is option A is the correct one. It's called as the Pasteur effect. That Louis Pasteur has actually discovered that when he was working with the yeast, both aerobic and anaerobic respirations. At the time he discovered, we call it as a Pasteur effect. Then what is that Emerson effect? Can anyone put it?
In photosynthesis.
Sorry for disturbing.
Yes, photosynthesis. Can anyone say the wavelength, teachers? Beyond
Yes,
which
sir.
wavelength?
380
Yes, yes,
photosystem.
yes, mainly into 680. Very nice one. Thank you. 680 and beyond. The observable red and far red, both that area, what effect produced when immediately exposing of this photosystem is called as Emerson effect. So Blackman's law we know already, and Chargaff rule, actually nowhere it is related here. It's related to this, as we know in that
DNA.
yes,
In pure
DNA,
intermediate, we will have such Chargaff rule.
yes, ma'am. Yes, thank you. So the correct option for this particular question is the option A is the correct one.
Pasteur.
Yes, Pastor's effect.
Pasteur.
Yes. Thank you, teachers.
Sir, excuse me 1 minute.
Yes, ma'am.
Sir, is it like is it from anaerobic to aerobic or from aerobic to anaerobic? Actually,
Anaerobic to
place
aerobic, ma'am.
anaerobic, sir.
When anaerobic organism doing the process of aerobic respiration, what happens is called as Pastor's effect. And there is no any effect for us aerobic to anaerobic. We have this lactic acid fermentation we have, that is only.
Sir, lactic acid fermentation is coming under, it is not switching over from anaerobic to aerobic, right?
No, aerobic to anaerobic.
No, that is what I have one, always I have one, what to say, unclarity. Like, it is actually not from anaerobic to aerobic. Actually, from aerobic to anaerobic only, no, sir. That is what I actually have a doubt.
You're asking about this lactic acid fermentation, ma'am?
Yes, sir. Actually, when we start the initial process, we warm the milk and then we put it. That time, actually, we are having aerobic condition.
Aerobic,
And once
yes.
we close
Yes.
the lid, it goes to anaerobic. So I actually used to have such a sort of, I do not know whether it is correct or wrong, but usually I have to have this confusion.
Yes, ma'am. Actually, here it's depending on the bacteria, whether it is an obligate anaerobe or it's an aerobic facultative anaerobe, ma'am. So based on that only, it is decided whether it is an aerobic respiration or anaerobic respiration is going to follow. If it is an say for example, methanogens, if you take it in, however you keep it outside, that aerobic respiration is never going to take place in that. Only anaerobic respiration, even if you supply enough oxygen, it is not going to convert. It is not
Yes.
going. Yes, ma'am. Yes, ma'am.
Archibacteria, they are.
Yes, ma'am. Yes, that's what I was talking about. There it will
Sir.
be a obligate anaerobe. Whatever we do, there is no possibility of undergoing aerobic respiration.
In that case, it is okay, sir. But
Yes.
here only I have a doubt all the time.
It's a facultative. Yes, ma'am. Facultative. So
actually it starts with aerobic condition and only then it will go to anaerobic condition. Whether it is lactic acid fermentation or it is alcohol fermentation, actually it starts from aerobic and only then it switches over to anaerobic due to facultative ferment. That
yes,
is
yes,
facultative.
yes, 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 ma'am. Yes, ma'am.
Actually, this is what always I used to have some uncertainty always. That's why I just
Okay.
want clarify.
Okay, okay. Now hope you are clear with that. Thank you. So this is that lactic acid, alcoholic, and aerobic. All the three different. The pyruvic acid form the end product of glycolysis. It has the fate depending on the need of the cell and what type of the facilities are present in the cell. Based on that, the fate will be different. It will differentiate. So lactic acid fermentation also may take place, or alcoholic fermentation. Both are in anaerobic condition, and in aerobic respiration also may take place. So if it is fermentation, both anaerobic respiration, which alcoholic as well as lactic acid fermentation, we need to very well highlight the role of yeast, and yeast, how it is happening in the muscle cells, and the lactic acid bacteria there and all, how it is happening. We need to, with a given example, the student must know. Where the students are making again error is when writing the flow chart. So this flow chart must be explained in a very, the students must explain it very well in the exam, because the exam question will also be based on only the flow chart. So they have to know, they need to be practiced in this regard. Here we need to give marks, say for example, how they are labeling correctly, the utilization of energy, how they are labeling, whether they are writing here itself, what is the net gain of the ATP if they are highlighting. Then based on that, we can give the marks. And if they are not writing, we can reduce the marks and we can make them understand the importance of writing all this labeling, so that students will have the clarity how to write the flow chart and what are the correct steps must be mentioned in that. And also the 13 students will have definitely in the 12th standard and all, this concept itself is there, but in 11th they will have definitely doubt that the yeast put itself poison to death when the level goes beyond 13%. So if it is so,
Yes.
there is a question in the screen, you may be able to see. You can try answering that. You can put it in the chat box also, teachers. Or you can even orally say. Can anyone say the answer for this?
Sir, actually, yeast can tolerate, I do not know the concentration. They can tolerate alcohol concentration. When higher the fermentation, lower will be the survival rate of the yeast.
Yes, ma'am. Yes. But whatever the level we increase, the fermentation level we increase, the 13% beyond that percentage is calculated based on how much over the concentration of the fermentation we take, the
Yes.
broth, whatever we take. So 13% beyond, if it is there, definitely it will put that yeast to death. It will kill. Even if it is 14%, that definitely yeast will die. Then how
Yes.
come there are beverages having more than 13%?
Enzymes are added with that, no, sir.
Yes, ma'am.
Because of the
Okay. Yes, anybody can add on to that? Actually, the yeast concentration is increased by the distillation process only. The while production, it will be maximum 12 to 13% only when the fermentation gets over. At the end of that, the percentage alcoholic concentration will be hardly 13% only. The concentration will be increased by the chemical process only, not by the biological process. That is a chemical treatment in which by distillation, the percentage will be increased. So that is the students will have doubt. We need to hear itself. We need to highlight that particular topic, because my students have asked that question. So they had that doubt, what will happen that beyond 13%. So we need to hear itself. We need to highlight it. And enzymes, again, the role of various enzymes played in this alcoholic fermentation, as well as the lactic acid fermentation, they need to keep in mind. So those enzymes are very, very important for the children. And we can even elaborate the role of the enzyme, how this enzyme is functioning. And here only 2 enzymes for alcoholic fermentation they are going to learn, and this another one, lactic acid, only one they are going to learn, so that we can explain. And also then a muscle cell, what happens here itself, if we highlight the role of white muscles and red muscles and all here itself. So this is the things we need to highlight to the children. Next one is aerobic respiration. Here again, I would like to show a video. Teachers, please you make use of this. And let me play the video to you. Just see whether you are able to see the video.
Sir.
Yes, teachers. So when we say they show this video, students will have some idea like this, any video, but mostly it's a very nice video. I have downloaded from an official website. So usually I do show so that children will have a clarity on that exactly the cellular level, how it is going to take place. Here they will have a confusion about where the matrix, which side matrix located, where that cytoplasm is there, where the proton gradient is created. They need to be very clear about that proton gradient. Even if you take the NEET question paper of last year when I have checked, you can see the questions asked based on that. So there is a need of clarity about where this proton gradient is created, what are all the events are going to take place in the matrix, and what are the events are going to take place in the membrane system, what are the things are going to happen. They should have a very clear cut differentiation between these two. There we need to really give a more focus so that children will be able to, without any doubt, students will be able to continue in that. And also the 2 steps about that, as we already said, the matrix of mitochondria and also in the inner membrane, what are all the things are going to take place, how the stepwise removal of that all the molecules are there happening, and same way in the inner membrane, how the passing of electron will lead to the creation of the proton gradient. Both the students must be able to differentiate very well. Usually I will say I won't teach this link reaction before itself teaches. I will directly straight away enter into the TCA cycle. And students will have the doubt, how come I will usually I will ask them to ask questions. How come that a 4-carbon molecule binds with the pyruvic acid lead to formation of a 6-carbon molecule, that is citric acid. Because pyruvic acid is a 3-carbon molecule. 3 plus 4, it should be 7. What happened to that 1 carbon? Student will be asking this question. So we need to give a scope for the children to ask such a question. Even if you see the discovery, that citric acid cycle was discovered much earlier. After around 7-8 years only, that particular link reaction was discovered. They had no idea about that acetyl-CoA. They have discovered much later, after the discovery of citric acid cycle. So that we need to make the children to question. That question only will come from the mind of the children when they understand the number of carbon atom correctly, and the stepwise reaction, if they understand only, that question would definitely come to their mind. So here we need to highlight that pyruvic acid straight away will not enter into the Krebs cycle or citric acid cycle. It has to be converted into a 2-carbon molecule by the decarboxylation step, that we need to say, and it forms acetyl-CoA. Can anyone say what is that A stands for, teachers? Can you say what that A stands for?
I just know that only as an coenzyme A.
Yes, ma'am. Yes, very nice. Yes. You can try anybody else. What is that A stands for? Very simple. The scientists had no option, so he gave the name A. That A is actually activation. He gave the name activation, that he simplified and he gave it as a A. So it's activation. This enzyme is activated so that a citric acid cycle can take place. So A actually stands for activation only. So pyruvic acid. Yes, ma'am. Yes, ma'am.
Like it stands for activation, you said. Actually, what is that highlighting? A is for activation. What it is going to activate?
Now I'm going to say that, ma'am. Yes, very nice. Now I'm going to say that how the scientists have discovered, then it will come to know.
Okay.
Now pyruvic acid with the magnesium, here also the cofactor very important. It will, in the presence of pyruvic dehydrogenase, it forms the acetyl-CoA along with that carbon dioxide. And that reaction they must write very neatly. The 2-carbon they must clearly keep in mind. It's a 2-carbon molecule that they must be able to keep in mind. This 2-carbon molecule, then it is going to enter into the cyclic process, where the 4-carbon molecule, when it binds, no need to reveal that 4-carbon molecule immediately. We need to say the 2-carbon molecule plus binds with some molecule, that X molecule, it gives the answer 6-carbon molecule, which is a citric acid. Because of this, we call it as citric acid cycle. All this after revealing. Later, we can reveal that the 4-carbon molecule is nothing but when the end of the cycle, when everything citric acid, that ketoglutric acid, all this thing completed, at last, when it becomes a oxaloacetic acid, there we need to reveal that this is the 4-carbon molecule. So the continuously the reaction, how it is taking place, we need to explain in that way. The students will keep in very mind for a long time. So here also, as we already said, the stepwise removal of carbon dioxide, we need to make the children to memorize. We need to make the children to keep in mind. Then only the students will be able to write it very well. So again, stepwise marks can be given for this particular topic. Each and labeling correctly. If they are labeling, we can give the marks. So this and all we need to take extra care. Now teachers were asking one teacher, what is that activated here? Now we are going to reveal the answer for that. So first before that, both are there in the same paragraph, same page only. So first, when the Krebs was discovering this, Hans Krebs was discovering this, a biochemical, is a very nice scientist. He was much interested in biochemistry and he wanted to know the chemical reaction that is taking place in the living system. When he was discovering with the same way what we said, citric acid, he first identified a 6-carbon molecule. Then only he had a doubt that how come a 4-carbon molecule bind with the pyruvic acid to form a 6-carbon molecule. There is some reaction taking place, that was he said, but he was not able to find what is that answer. That acetyl-CoA reaction he was not able to discover. Then in 1945, much later, that is the time when Fritz Lippmann, that is another scientist, he only discovered that the link reaction. Ma'am asked what is activated here. The acetate is activated, ma'am. Can you note it down, ma'am?
Sir, what, sir? You have written
Acetate.
only coenzyme.
Ma'am?
Sir, I can see only coenzyme. Actually, what is activated?
This acetate is activated into acetyl CoA. So acetate is activated, so we call it as acetyl CoA.
1 minute. Actually, pyruvate gets decarboxylated and it becomes acetyl, right?
Yes, ma'am.
Pyruvate is getting decarboxylated, only then it is getting converted into acetyl.
Yes, ma'am.
Acetyl is a 2-carbon compound. And
Yes.
for that, coenzyme A is helping.
A is
And,
helping for that. Yes.
then why A, sir? It is just only a cofactor, right?
The acetate, acetyl CoA, I am asking about that enzyme acetyl CoA is there. What is the name of that A? I am asking, ma'am. The A, what does that A stands for? Means the previously the enzyme will be in that before activation, that A enzyme will be activated. So we call it as acetyl CoA. That A is nothing but stands for activation. That is the only thing. A for activation. When students are asking what is that A stands for this activation. Very nice.
Okay.
So next one is the same thing, the stepwise removal, stepwise decarboxylation. We need to explain to the children. So step by step, what is happening? What is that acetyl? Directly, as we already said, directly no need to reveal that 4-carbon molecule, that oxaloacetic acid, no need to reveal in the beginning itself. Let us directly start with citric acid. At last, we need to reveal it. Then students will be able to understand the number of carbon atom numbers present there. That and all students will be able to understand. Then the TCA cycle in an elaborated way. It's a very important topic for the children. And this is the elaborated TCA cycle. This cycle we would have studied, but students sometimes those who are very toppers in the class and all, they come with a lot of questions, they refer some book and they learn all the steps and they come and ask us. So there also, even the NEET and all, the elaborated questions are asked. So in that way, we can also explain this particular the elaborated cycle. This is the original cycle given by the Krebs. So this also we can explain to the children. Shall we go to the next one, teachers? So next one is a very important and complicated topic. You can put down what are the challenges you face during explaining this one, ETS, while explaining ETS.
Sir, I have never won the students' mind in this regard, because even if I show this when I download any video from YouTube, these
Yes.
children
Yes, ma'am.
cannot correlate what only we are showing, because they are in different shapes and size.
Yes,
And
ma'am. Yes.
when children sees that, they
Yes.
don't understand, or they are unable to correlate what we are showing. And
Yes,
I have
ma'am.
never said so far.
Exactly, ma'am. That is true. That
And
is
if
true.
you can tell me one easiest method to make these children understand, this will be of great use to me.
Definitely, ma'am. Definitely. Yes. So electron transport system, what my students where I have analyzed in these many years, where the problem they have understanding and how come that be sorted out. First, I have understood this. The students need to understand the structure of the inner membrane, where that with respect to the inner membrane, where the matrix is located, which side the cytoplasm is located. First, the students, some amount of clarity students will get when they are clear with the location of this particular inner membranes. Of course, as ma'am said, even if you show the video, in video the location will be somewhere else. If we show the different materials, if we show in that every material, the location would differ. The first area where we need to explain the students in a very clear, and the students must have clarity where the inner membrane is located, which side the matrix is located, which side that inner membrane space is there. If the students are very clear that the proton gradient is going to be created in the inner membrane space, it is more or less similar to this what we have studied in chemiosmotic hypothesis. But they must know that proton gradient is going to be created in the inner membrane space. That after that outer membrane will be there. From the outer membrane, it is a cytoplasm is there. Now it is arranged in such a way that we need to explain children in the inner membrane, outer membrane, in between, that is the area where proton gradient is going to be created. And ATP, I never start the concept with the complex 1 or complex 2. I always start this topic with the complex 5 only, teachers, because student first of all must know where the which side is ATP is produced. In the chemiosmotic hypothesis, the ATP is produced towards the stromal side, but here the ATP is going to be produced towards the matrix side. Then the proton gradient will be created just opposite to that, that is in the inner membrane space. That clarity, if the students are having the beginning issues, whatever the issues they have in understanding of the different locations and all, I believe they will be more clear about it. So ATP directly, instead of starting with complex 1, complex 2, complex 3, of course there and all electron transport is going to happen. That is, we'll see when we are explaining about the ETS, we'll go with first onwards. But while we are explaining about the structure of it, then we need to better explain about the complex 5 straight away, and where the ATP production is going to take place, where it is going to be stored. So opposite to that, where the hydrogen proton gradient is going to be created, if we give this clarity to the children, I believe more than even 50% student will be very clear with this ETS, electron transport system, I believe. What is your opinion, teachers? Do you think so? Will it happen? Mostly students have the doubt of, they relate it with the chemiosmotic hypothesis. ATP is produced outside from the thylakoid membrane, outside towards the outer side it is produced. Students will have that same mentality. Here when it comes to mitochondria, they need to understand it is going to be in the matrix. So they have the problem with understanding of where is the matrix, where is the cytoplasm, where the inner membrane space, and where that the inner membrane is located. If they are clear with this particular regard, with this particular location, mostly around most of the things they will be very clear. Then when we start with the electron, stepwise electron acceptance, when we are going to link, definitely the complex 1 is NADP dehydrogenase. They need to memorize the words complex 3, complex 2, then complex 4. When we are explaining that, they need to understand from that the molecule, one atom, from that they are going to remove the electron. Electron is going to be stepwise removed. At the time, proton will be pumped into the inner membrane space. The proton is going to be pumped into the inner membrane space. That is a very important keyword they must keep in the mind, so that how the proton gradient is created, they will be able to understand. And mobile carrier elements, like both the mobile carrier elements, they must be very clear, and that will help them understand how that electrons are transferred from one photosystem, one complex to another complex, so that it leads to the proton gradient increase. The increasing of proton gradient is the high weightage has to be given. Then the electron transport. Electron is transport, at the time only proton is going to be gradient has to be created in the inner membrane space. That clarity student must have while learning this particular ETS. And at last, the proton gradient is created, that leads to formation of this particular ATP synthesis. The fifth complex is going to use those gradient, that electrochemical negativity, and that gradient, using that potential energy, that ATP synthesis is going to happen. Here, what is the difference they are going to learn from, which is chemiosmotic hypothesis, and here here not only just ATP is going to be produced as it is, there are other energy carriers. Those energy carriers, from that how they are going to be conducted or going to be converted into ATP. That balance sheet is going to be a important factor we need to explain in this particular topic. So the structure of ATP synthesis enzyme, already we said this, we need to explain in the previous lesson itself. When we explain there, there is no need of explaining this particular here, but only the location they have to know where it is located. So this particular topic we need to explain in the previous lesson itself. When we are explaining about photosynthesis in the dark reaction itself, we need to explain this particular dark reaction, the light reaction. When the chemiosmotic hypothesis happening, there itself we need to explain the role of F1 and F0, this integral membrane protein and the peripheral membrane. We need to explain then and there itself, so that topic also they will be able to understand very well. This topic also they will be able to understand. So next one is the entire structure. We can show the videos to the children. Now if we show the video, if we show any other learning material or any teaching aids, students will definitely will be able to understand it. They will know what exactly that this electron transport system is all for. It can be very well related to chemiosmotic hypothesis. I do definitely do that. The chemiosmotic hypothesis, it has to be linked. It has to be understood. The role of proton gradient, the role of ATP synthase. These two has to be given highlight, and the other complexes and all, how the different energy carriers are going to be playing a role, which hydrogen, that and all is a source from which hydrogen is pumped into the inner membrane space. Otherwise, how the hydrogen will be there. So students need to understand how that hydrogen from various energy carriers, how that hydrogen is pumped into this particular inner membrane space. They need to be very clear. Then the net gain, the balance sheet, we need to explain to the children totally how many ATPs are directly utilized or synthesized. Utilized also we need to say. Then during the link step, what is happening? Then during the TCA cycle, how it is happening. So all together, when it comes 38, already 2 ATPs are utilized. That also has to be, students must be clear about what happened to the 2 ATP. And last, third, this and all not elaborated manner given in the book, our reader alone. So some of the areas we need to actually highlight, put our input and make the children to be clear with this particular topic. And the net gain, how that net gain we reached, they must know. Then that difference between aerobic and anaerobic respiration, that all very simple, they know very well. Only in the terms of ATP, they may not know in terms of ATP, the gaining. That alone we need to explain to the children, so that they will know what exactly is that difference between it. Otherwise, they will simply write like aerobic respiration takes place in the presence of oxygen. Other one is in the absence of oxygen. The what they have learned in 10th class level, the same thing only they will be continuously repeating. That we need to change in the children. They have to learn about the other some extra points they must know. So the amphibolic pathway, here again, when we are explaining this, we need to again go back to the glycolysis cycle, and we need to say that glucose is definitely a very important respiratory substrate, mostly very important, but doesn't mean that, because students have right from the small class they are studying only the carbohydrates and fats are the energy giving foods, as if we don't get any energy from the other substance. They don't learn, they don't know that. So here we need to highlight, from all the molecules have the ability to give energy, but the only job of the carbohydrate is to give energy, whereas the other molecules, they have some other work also to be performed in our living system. So it is not an amphibolic pathway where we need to highlight. It is not all exclusively a catabolic process, where anabolism also occurs because of the various molecules are either get taken into the glycolysis or withdrawn from the glycolysis. By anabolism and catabolism, both it is done. So we call it as an amphibolic pathway. And we need to highlight, if it is amino acid, generally it enters into the cycle of aerobic respiration or respiration in which level. It directly converted into pyruvic acid, they must know that. Same way in the fat and all is converted into the glyceraldehyde. So that also we need to highlight to the children. What are the level it enters into the respiration, that we need to definitely highlight. The next one is the respiratory quotient. The students will, they find it very easy only. Usually they learn it very well. What is the respiratory quotient of different molecules? Beyond that, we can even give some examples from net or internet based some examples also we can give to the children. So we can give more to find what are on their own, they have to find how many molecules are used. For this, definitely the prerequisite is equation is very important. We have to give how many oxygen is produced and how much carbon dioxide is utilized. They must know that how much carbon dioxide needed to be produced and how many oxygen needs to be utilized, that they must know. Then only they will be able to correctly calculate it. Like, already the tripalmetine, like more examples we can give to the children so that they will be able to find the respiratory quotient of various molecules, including different type of protein. If you take a fat, not all fat is going to have the same 0.7. Some fat will have even 0.5. Some fat may have even 0.9. So we need to give various examples to the children which has that what. Same way the proteins, the average is 0.9. There are some protein, it may give much lesser than that also. We need to give some examples. Even net-based example is okay, though they need not to study for the exam, but they must know. They must know how to calculate. By doing that, we will be able to do that. So that's all, teachers.